pleasure to introduce you all with the speaker of today's session, which is a very great initiative from the side of Gopal Jee sir. That is a science dialogue series. And as far as I am, since the first episode of this science dialogue series, I am associated with it either as a listener or as an orator or as an attender. In all three forms, I have attended these science dialogue series, and believe me, this is one of the best series that I have been attached to, and that is really imparting great knowledge to the students or to the faculties or to anyone who is trying to get some useful information, some relevant or the recent information in different fields of the science. So it is my pleasure to introduce you, Ashwini sir. I remember he was the second presenter of this science dialogue series, and his topic was on first.
First,
Okay. He is the first one. The topic was on tissue engineering.
Yes.
And that was really very good topic. Really very good presentation, because I then interacted a bit with the sir that my own MSc dissertation was on tissue engineering. So everything was getting rebraced in that time frame. So thank you, sir, first of all for that series. And now it's time to introduce you for the current session of this science dialogue series to impart the knowledge on biotechnology and the role of artificial intelligence. Recently, we all have been in terms of, or we all have been in contact of this term, which is artificial intelligence or AI. Now, the question is that how this artificial intelligence, it can be implicated or it can be linked to the biotechnology. So the topic of today is very intriguing. It's very interesting. And talking about the speaker, he himself is a very interesting person as a you can say as a researcher or as an academician. So Dr. Ashwini Kumar, he is presently working as an assistant professor in the Department of Life Science, Sharda School of Basic Science and Research, Sharda University, Greater Noida, Uttar Pradesh. Previously, he has served as an assistant professor in the field of biotechnology, bioinformatics at NIIT University, Neemrana, Rajasthan, at the Department of Biotechnology as IMS Engineering College, Ghaziabad, and as a lecturer in the Department of Biotechnology at NIT Raipur, Chhattisgarh. So he has a very vast experience as an academician. He has dealt with a lot number of students. He understands the recent requirements, what is required to get incorporated in different fields of biotechnology. That is why he is here to present us the topic of this biotechnology and AI. Regarding his research experience, he has a lot of research experience. One of the topic that he has already discussed us with the first science dialogue series in the field of this tissue engineering. More of his work is concerned about the biomaterial science, more specifically to the field of novel drug delivery system. The target application of his research are formulating the non-invasive or minimally invasive drug delivery approaches for the better result, both in terms of pharmaceuticals or related to the patient compliances. He also has interest in the field of diabetes, how it can be tackled with the help of the tissue engineering. And regarding his accreditation, so his research accreditations are very enormous. He has authored 23 journal articles in national and international journals, 8 book chapters, and he also has 1 book in his credit. And also regarding his work from the PhD, he also holds a patent on his work. So this is a very brief introduction about the academic and research accreditations of the today's speaker, Dr. Ashwini Kumar. So I hope that this topic of today may be very intriguing, may be interesting to all the participants who are here to get the benefit from the thoughts or from the presentation of Dr. Ashwini Kumar. So, sir, now our participants, now our other members of this science dialogue series, they are up to you. And now this presentation is I am handing over to you. So, sir, please.
Thank you so much, ma'am, for this very kind introduction. And if you and Gopal sir permits me, I can switch off the video to have uninterrupted network.
Yes.
And I'll just be sharing the presentation. Yes. Sir, is this visible?
Not visible. Yes, now visible.
Okay. Right.
Full
So,
screen. Full screen में कर लीजिए.
sir, I made it full screen. Is it visible, sir?
नहीं, स्लाइड शो मोड में करिए. स्लाइड शो का वो क्लिक नीचे.
सर अभी ठीक है?
नहीं, मुझे थोड़ा लग रहा है. एवरीबॉडी, इज इट विजिबल? दिख रहा है, लेकिन वो स्लाइड शो का एक होता है ना, यहाँ नीचे देखिए आप, फॉर स्लाइड शो मोड फुल स्क्रीन आएगा.
सर हम फिर से करके देखते हैं. सर अभी ठीक है?
अभी तो दिख ही नहीं रहा है.
सर एक सेकंड. सर अभी दिख रहा है सर?
हाँ, दिख रहा. चलिए करिए. हाँ.
जी. Okay, so firstly, welcome everyone for sparing your time and giving me the chance to interact with you on this very interesting topic, biology and artificial intelligence. So we all know that biotechnology is made up of two words, basic words, biology and technology. In fact, we are using a lot of technology from every possible field in the field of biology, and that's why biotechnology came into being. But recently, there is a lot of discussion on artificial intelligence, and one very interesting aspect of incorporating artificial intelligence is in biology again, and that's why I chose the tagline of the topic that it is another technological marriage between biology and artificial intelligence. So let's begin with some of the basic sciences. So the left-hand side picture is very well known to everyone, I hope. It's about DNA, and the picture of these beautiful scientists, I would just again kind of recall for everyone. Is this slide visible?
Sir, your slide is not moving.
Yes, फर्स्ट स्लाइड सिर्फ दिख रहा है, एंड स्लाइड शो मोड भी नहीं है.
Sir, just a second. Let me. So I think इसका कुछ issue है.
मुझे भी मेल कर दीजिए आप.
Sure, sir.
यस, नाउ.
सर हम सर व्हाट्सएप पर आपको भेज दें?
नहीं, मेल में ही करना पड़ेगा.
मेल कर दें. जी सर.
जीमेल वाला जो है.
जी. आई थिंक लैपटॉप कुछ इशू है. Extremely sorry for this technological glitch. So technology is always a two-edged sword. And a lot of problems also. ये सर हमने मेल कर दिया सर.
जीमेल वाला ना?
जी सर.
नहीं आया. जस्ट जब इंटरनेट स्लो रहेगा तो आपका पीपीटी आने में भी टाइम लगेगा.
जी.
डिलीवर हुआ?
यस सर. यहाँ से तो
हाँ आ गया.
यहाँ से दिख रहा है सर.
हाँ आ गया. यस, आई एम डाउनलोडिंग. Okay, so now So now I am presenting, है ना?
Sir.
Is it visible?
Yes, sir.
So now you can start.
हाँ सर,
सबको दिख रहा है?
स्लाइड शो
Slide.
सर दिख रहा है वो स्लाइड शो वाले पर जी सर.
Yes.
Yes, yes.
Yes, sir. Thank you, sir. So, yeah, I was just beginning this session by saying that we already are using a lot of technology. In fact, all of the technological interventions that is possible, and attaching it with biology to so that biotechnology can function. And in last few years, artificial intelligence, which is completely a core computational platform, is being attached to biology, and that's why I made this tagline that it's another technological marriage between biology and AI. Sir, next slide.
चेंज किया मैंने नेक्स्ट. दिख रहा है नहीं दिख रहा है?
Yes, sir, visible.
आपको नहीं दिख रहा क्या असली?
सर हाँ जी सर, ठीक है सर. सर हम सर हमें इसमें नहीं दिख रहा है, बट हम अपना पीपीटी भी खोल के रखें. सो जैसे ही वो खत्म होगा ask you to change.
Okay.
जी सर. Okay, so the first slide of this is about the basic science. So I hope everyone is familiar with the DNA, and just to make everyone recall about these interesting and beautiful scientists. So everyone has heard of Watson, Crick, and Franklin, and they were the pioneer in deciding and deciphering the structure of DNA, the double helical structure. Moving on to the right hand side picture of Arthur Kornberg. So he discovered the DNA polymerase enzyme, and this is the set of enzyme that we study in DNA replication. Coming down, we have Arbor, Nathan, and Smith, and they discovered a very interesting enzyme which is called restriction endonuclease that cuts the DNA chain or any nucleic acid chain from in between, not from the terminal end. Moving again to Dr. Hargobind Khurana. So by birth he was an Indian, but he performed all his studies and research abroad, and he was awarded Nobel Prize, I think in 1964, for decoding the DNA, the triplet. Moving on to the last picture is that of Carey Mullis, and he's the person who invented the technique called PCR, polymerase chain reaction, which was a huge technological advancement for application of gene right from the conventional cloning experiment. And a very interesting fact is that mostly the Nobel Prize is awarded for the basic science, and even though the PCR was a technical advancement, Carey Mullis was awarded a Nobel Prize for this technique. Sir, next slide. Yeah. Now it was a very basic of chemistry and biology, and utilizing all those concepts, the technology came into being. So if we can see the picture on the left hand side, we are all familiar with this, and this is the favorite topic of Gopal sir, also recombinant DNA technology. So in case we want to have a product from a gene of interest, we take the gene of interest from the longer piece of the DNA, cut it down using a restriction endonuclease. We take a vector, most commonly by the name of plasmid or cosmid. We cut the vector with the same set of restriction endonuclease, combine the gene of interest and plasmid, and this new recombinant vector is then inserted into a suitable host, so that as the host divides, it also translates the gene of interest. Once we have a lot of cells, the host cells, we cleave the cells and take our protein of interest. So this is the technology which was again discovered by Paul Berg and Boyer and Cohen. So this is how science leads to technology always. And I always say in my class, technology is the child of science. So if there is no science, there won't be any technology. Sir, next slide. Yes, and this is one very interesting picture I always use. The top picture, the picture at the top, it was taken in 1951. So you can see almost 10,000 pounds of pig pancreas makes around 1 pound of insulin. So everyone knows that insulin is kind of lifesaver for people who are suffering from diabetes and they don't respond to the oral medicines. Currently, though, we have all the insulin in the world made through recombinant recombinant technology. But at that point of time, in around 1951, though insulin was discovered in 1921, but till 1981, for almost 60 years of age, it was the crude insulin extracted from the pancreas, and that was delivered to the people suffering from type 1 diabetes. But now, if you can see the picture downwards, we have these sorts of.
हेलो हेलो क्या हुआ? अश्विनी जी, he left.
Maybe some connectivity issue.
Calling him. आप डिस्कनेक्ट हो गए थे अश्विनी जी. हेलो, मेरी आवाज आ रही है अश्विनी जी? हेलो,
हेलो,
आवाज आ रही है आपको? मेरी आवाज आ रही है? हेलो
हेलो.
हाँ, मुझे आवाज आ रही है. आपको आ रही है मेरी आवाज?
हाँ जी सर, जी सर, जी सर, जी सर, जी सर.
थोड़ा नेटवर्क देखिए. आप एग्जिट हो गए थे. नेटवर्क देखिए थोड़ा सा.
सर हम जी सर, जी सर, हमने दूसरा नेटवर्क चेंज कर लिया सर.
ओके ओके. इज इट विजिबल?
जी. यस सर. सर नेक्स्ट, सर यहाँ से डिस्कनेक्ट हो गए थे.
हाँ, यहाँ बताया था आपने. इसका नेक्स्ट यहाँ से कर.
ओके. जी जी सर, जी सर. So, sorry for again that technical glitch. So, not only insulin, we have a lot of recombinant products which are currently in the market as medicine. We have around hundreds of those recombinant products. And not only in medicine, this genetic engineering or genetically modified organisms are there in agriculture field also. This picture on the right hand side is that of a BT cotton. So very recently, a genetically modified mustard has also been approved in India, for which the research was going on for last 20-25 years, and that is from the lab of former VC of Delhi University, Professor Deepak Pendil. Okay, sir, next slide. Yes. So apart from all these recombinant products, we have a lot of vaccines also. So the left hand side picture you can see is that of different types of vaccine. The conventional vaccine though consisted of live attenuated vaccine, but currently we have a lot of recombinant vaccines. So if you are aware that during this COVID, Covishield was one of the most used brand in India. Covishield is actually a recombinant vaccine. Apart from that, we have subunit vaccine where we only take the gene of interest that can encode a protein, and that protein will be considered or recognized as antigen in the body. So we have subunit protein also, which is again a part of the recombinant DNA technology. Apart from these kind of novel vaccines, we have a very recent therapeutic achievement which is called CAR T cell therapy, which is currently approved for blood cancer. Worldwide, currently we have 6 CAR T cell therapy which have been approved initially in the initial phases, but because of their extremely high cost, they were not available in India. So the first 6 CAR T cell therapy were almost of the cost of 3 to 4 crores of rupees for single shot of injection. But with the indigenous technology right from the scratch, Professor Rahul Purwar from IIT Bombay, Department of Bioengineering, IIT Bombay, developed a completely indigenous CAR T cell therapy which has recently been approved in India, and that costs around 30 to 40 lakhs of rupees. So the cost has drastically came down. In fact, the second CAR T cell therapy, completely indigenous in India, has recently been approved, which it is from a company called Immunil, which is funded hugely funded by Biocon. So these are some of the interventions that we have through recombinant DNA technology. Sir, next slide. So, moving on to the next paradigm of biomedical engineering, or kind of, it's a no man's land between biotechnology and biomedical engineering. So, this is just a small slide of tissue engineering. It's kind of creating artificial organs and tissue right in the lab so that they can be used for implantation and transplantation. Apart from the conventional hypothesis of using them for implantation, these artificial organs or tissue can be very well used in drug discovery, replacing the conventional usage of animal models. So, next slide. This is just an example. You can see 3D printed windpipe gives infant breath of life. So it is not engineered tissue. In fact, it's a artificial windpipe which was designed by 3D printing, and it was surgically implanted into a patient, infant patient who was who was diagnosed with a developmental disorder and was unable or found very difficult breathing. So this windpipe, artificial three-dimensionally created windpipe, was implanted in that person. So next slide. So if you can see the picture of this artificial eye, this is a paradigm directly from biomedical engineering. It has nothing to do with biotechnology. This area is called bionic, and we have currently bionic eye, bionic hand, bionic legs. In fact, there are not a lot of bionic nerves being experimented upon. These are completely electromechanical instruments that can be implanted in a person who requires this. So in case a person is completely blind and there's no scope of eye transplantation, these kind of bionic eye can be implanted into that person. Okay, so in a very short, I've just tried to discuss what are different areas directly from biotechnology or from the no man's land of tissue engineering or from directly biomedical engineering that are currently at the rescue of humankind. But sir, can we have the next slide? Yes. So this slide is, so where is the problem? If everything is going fine, we have a lot of recombinant products as a medicine. We have a lot of products coming from tissue engineering. We have directly electromechanical instruments in the form of bionic. So where is the problem? Sir, next slide. Yes. So one of the problems, sir, I'm still in the previous slide.
Why is protein folding important? Yeah.
No, sir. The previous slide. Sir, previous slide. Yes, sir. So one of the big, sir, after this. Yes. So one of the problems, a very basic problem in biology is that of protein folding. So every one of us, I hope, knows that every functional protein has a very definite structure, and that is thermodynamically stable. In fact, everything that has a function, the function is because of its structure, whether it's a small molecule or it's a protein. Now, the problem is that the gene sequence can give us a very good idea of the amino acid sequence. That is the primary structure of protein. But every protein, every functional protein has its normal function because of its folding. Whether the folding is in a secondary state or a tertiary state or a quaternary state. But the function is always because of the structure. Now, this is called a problem because it is extremely difficult currently to experimentally predict the correct folded structure of a protein. If anyone of I hope, because this is a very common disease, the Alzheimer's and Parkinson, these are some of the very common examples of how can a misfolded protein result in a pathological condition. Now, this protein folding problem is again a problem because out of millions and billions of randomly folded structure that a single chain of amino acid can go in, it just stops at the most thermodynamically stable structure. And sir, can we move to the next slide? And a very famous scientist, Linus Pauling, who received, I think, 2 Nobel Prize, he was the first to predict the alpha helical structure. So he famously told that I think that we shall be able to get a more thorough understanding of the nature of disease in general by investigating the molecules that make up the human body, including the abnormal molecules, and that this understanding will permit the problem of disease to be attacked in a more straightforward manner, such that new methods of therapy will be developed. This saying is, in a very nutshell, is that unless until we are sure of the exact structure of the protein that we need to target in terms of drug targeting, we cannot design a drug for that. Because unless until we have a proper structure, proper mechanism of the disease known, we can't go forward with discovering the drug. So drug discovery, we all know it's a very, very long process. On an average, it takes almost 10 to 15 years for a drug to become a medicine. But what happens before that, hardly anyone talks of that. So unless until we completely know in and out of a disease, and more specifically about the a single disease can have multiple different targets. So more precisely, we have to know the proper structure of the target protein, and then only we can proceed for the drug discovery process. So next slide. Okay, so this slide is a very basic difference between a drug and medicine. So any molecule that can show a therapeutic effect is a drug, and when it is formulated in a particular manner, it becomes a medicine. So the structure that you can see is that of a paracetamol, and this is a just representation of a tablet strip. We are very familiar with Dolo and all these things. So paracetamol becomes Dolo. That means a drug becomes a medicine. Now, in this complete journey of drug discovery, it takes almost 10 to 15 years of time. It starts from early drug discovery, right from target identification and validation and discovery of the hit. And then when we are sure that, yeah, this particular molecule or this 10 molecules are good to go for this particular target, then we proceed for the preclinical study. So from in silico to preclinical and then to clinical development, it takes a lot of time. On an average, only the human clinical trial takes around 5 years of time. After everything goes well till the human clinical trial, we again have to go through a lot of regulatory steps, and therefore a drug finally becomes a medicine after a lot of time, lot of people being involved, lot of cost involved in this complete drug discovery process. So next slide. Yeah. Apart from drug discovery, in fact, the base of any treatment lies in the diagnosis. So this is a very basic fact, basic slide which shows the histological image of certain tissue. So if you can see, though it's written that the top one is of a normal tissue and the lower row is that of a cancerous tissue, but as a lay person or even a scientist who is not involved in these kind of studies, it is almost impossible to just see the histological slides and say which one is normal and which one is cancerous. And this practice needs a lot of time to do that, and therefore a pathologist or a clinical doctor who is specialized in cancer treatment can only say that this is a normal or this is cancerous. Otherwise, with a layperson eyes, it is almost impossible, or in fact impossible to say which is a normal histological tissue and which is a cancerous tissue. So next slide. Yes, sir. Again, the same thing. It is just a bit of enlarged image, so you can see left hand side it's a normal tissue, and the just below that it's an in situ carcinoma. Again, you are trained in that. Yes. So, sir, yes. Next slide, sir. So there's a very famous saying, to err is human. That means it is a very basic nature of human beings to make error. Now, in terms of drug discovery, there is definitely going to be a lot of errors, and even in terms of diagnosis, because again it's completely because of human intervention, there can be a lot of errors. So even a trained eye can be confused whether it's a normal histological slide or whether it's a cancerous histological slide. And just a minute problem or minute error with diagnosis can result in something very disastrous. So here is where artificial intelligence come to the rescue. So next slide. Yes. So artificial intelligence is a broad field which refers to the use of technologies to build machines and computers that have ability to mimic the cognitive functions associated with human intelligence, such as being able to see, understand, and respond to spoken or written language, analyze the data, make recommendations, and a lot of other things. Machine learning is an application of artificial intelligence that allows the machines, especially the computers and the programs associated with it, to extract knowledge from the data and learn from it autonomously. So in case of machine learning, in a very nutshell, the computer programs are being fed with a lot of data. They are trained to recognize certain specific features among all those data, and now to predict the input that we are giving as a fresh problem to the computer. So based on their based on the training with the help of those algorithms and the softwares, the computer can predict, suppose again with the problem of protein folding, a computer can predict the very exact folded structure of a very native or new protein. So next slide. Yes. So there is a very famous Leventhal paradox, and it says it would take longer than the age of the known universe to randomly enumerate all possible configuration of a typical protein before reaching the true 3D structure. Yet proteins themselves fold spontaneously within milliseconds. Predicting how these chains will fold into intricate three-dimensional structure of a protein is what's known as the protein folding problem. So you know everything happening inside a cell is within picosecond or femtosecond. And so Leventhal said that if we try to figure out the correct folded structure of a protein in vitro in the lab, it would take astronomical years to do that. And in the cell, the complete cell as a machinery does that within milliseconds. Among all the billions of possible folded structure, within millisecond, the cell decides what is the best folded structure of protein. So if we can somehow decide or predict this protein folding outside our cell with the help of computer algorithms, it will be a huge intervention in the field of drug discovery. So next slide. Yes. So one of the most used or talked about example currently in this protein folding problem is that of a software called AlphaFold. It was developed by Google. So there is a sister concern, or there is a button of Google called DeepMind, and DeepMind has developed the AlphaFold. So AlphaFold is an artificial intelligence-based system that predicts a protein's three-dimensional structure from its simple amino acid primary sequence. It regularly achieves accuracy competitive with experiments. So currently, as we all know, I hope everyone has touched the PDB website, Protein Data Bank. So all the structure present in PDB, if you can recall, if you can just after this session go and again check the PDB, the structures are there with the help of XRD or NMR. So currently, though we do have another advancement in the name of cryo-electron microscope, but currently all the structures in PDB are those predicted from XRD, X-ray diffraction, or NMR. But out of almost 200 million currently known proteins, we hardly know the structure through XRD and NMR of around thousands of protein, hardly. And again, this is where the problem still exists, that out of 200 or 200 plus million of proteins known through sequencing, we still don't know the accurate folded structure of those proteins. Most of the proteins that are there in PDB, they are either secretory proteins or the proteins present in cytoplasm, and more specifically, out of all the proteins or different classes of protein, the proteins which are embedded or attached to the cell membrane or nuclear membrane, to find out their structure is in fact much more difficult than finding out the structure of a secreted protein such as enzyme. So next slide. Yes. So this is one of the paper out of thousands of example. Currently, this is one of the paper from last year. AlphaFold accelerates artificial intelligence powered drug discovery, efficient discovery of a novel CDK20 small molecule inhibitor, and this is for cancer. So this is just one example how AlphaFold can accurately or very closely predict the structure of a target, so that after knowing the structure of the target, we can proceed for finding the drug for that. So next slide. Yes. Another very interesting example I find is of that of a company called In Silico Medicine. It's a Hong Kong based company, and it was obviously a startup. And why this is more interesting is that you can see the three divisions of this company. These are the three platforms, completely AI based platforms that it has made, In Silico Medicine has made. First one is Pandomics. It's a biology platform that helps the researchers to find a novel target for a disease. Another platform is Chemistry 42 for drug discovery, whether it's a structure-based drug discovery or a ligand-based drug discovery. And the third platform it has made is In Clinico, that predicts how a drug molecule is going to act in the clinical trial. So because, one of the problem with drug discovery is that almost 95% of the drugs fail to reach the market. There are a lot of factors. One major factor is that human clinical trials are there as a stage after preclinical trial or the animal trial. So a lot of drugs, it is widely reported, a lot of drugs which have shown good result in animal trial, they don't show similar results in human trial, and this is one of the results. They fail in different phases of human clinical trial. So this In Clinico platform developed by In Silico Medicine is going to again predict how a drug is going to perform inside the human body in different stages of clinical trial, according to different parameters that we judge. So next slide. Hello.
Yes, changed.
Yes, sir. Yes, sir. Yes, sir. Yes, sir. So this is again one example. If you can see the date below, it was published in 2021, around 3 years back, 2.5 years back. So In Silico Medicine initiates first in human study of ISM 001005, a novel drug discovered using In Silico's proprietary end-to-end AI platform. So the interesting feature of this particular company currently is that, as we know, that a drug right from the early discovery stage till coming to human clinical trials take around 6 to 7 years of time. But through all these three platforms, the Pandomics, the Chemistry 42, and In Clinico, using all these platforms, In Silico Medicine discovered a drug for a condition called idiopathic pulmonary fibrosis, and that particular drug with the code name ISM 001055 has reached human clinical trial within 18 months. So you can just think of the time it has reduced from 6 years to 18 months for a drug to reach the human clinical trial. And this is the achievement you can think of when we incorporate artificial intelligence with biotechnology. So next slide. Yeah, so this is just an animation for, Again, very recent achievement by In Silico Medicine. Again, they have again discovered a AI discovered molecule for a target called PhD for anemia. And in the picture below, you can see it has again discovered a drug for MYT1, and this MYT inhibitor is for one of the cancers. So this is how AI has come to rescue for drug discovery. So if we are using different AI platforms, whether for target identification, to understand the structure of the target, or to discover a drug right from the scratch, AI is almost everywhere. And as per a few reports, almost all the big pharma companies are currently using one or other different artificial intelligence platforms. So next slide. Yes. Again, one of the news. So, using artificial intelligence, scientists find a drug that could combat drug-resistant infection. I hope every one of us has heard of a term called antimicrobial resistance. That means microorganisms, especially pathogenic microorganisms, are gradually becoming resistant to almost all the present antibiotics available on Earth. One of the, I would use a Hindi word called Brahmastra. One of the Brahmastra antibiotic is colistin. And you can find over Google, there have been a lot of reports that a lot of pathogenic microorganisms are also becoming resistant to colistin. So just now imagine uh, if this AMR grows in the current rate, maybe within next 20 years there will be a time when even a simple infection can become fatal because there won't be any antibiotics left to treat any of the infection. And believe me, AMR or antimicrobial resistance is actually going to be the next pandemic. So this news from MIT says that using AI-based tools, scientists have found out a drug that could very well kill this particular pathogen, which is called Acinetobacter baumannii. It is one of the scientists have clubbed a lot of pathogenic or class of pathogenic organism under a class which is called escape organism. If I can just type it in the chat box, is ESKEPE, escape. So it's a class which consists of Enterobacteriaceae, Streptococcus, Klebsiella, Acinetobacter, Pseudomonas, and E. coli. So this is a class of pathogen which is extremely dangerous currently, and out of this escape class, one of the pathogen Acinetobacter baumannii was very well killed by this particular drug, which was not, I will say, discovered, but it was found out to be extremely effective to kill this particular pathogen. And you will be surprised to know that this particular drug was a already used anti-diabetic drug. So using artificial intelligence and machine learning, the team in MIT use a lot of drugs, currently approved drugs for different indications. They put those drugs as the input or training molecules in this machine learning platform, and out of all those molecules, the output was a very surprising anti-diabetic drug that was killing this particular pathogen extremely well. So next slide. Yes. Again, I hope you can recall in my earlier slide, I discussed about how correct diagnosis is very important. And when we have histological slides, it becomes impossible for a layperson without the relevant training to find out whether it's a normal histology or a cancerous histology. Now, artificial intelligence has come to rescue in this domain also. So you can see the title of the paper, Development and Validation of Deep Learning Based Automatic Detection Algorithm for Malignant Pulmonary Nodules on Chest Radiographs. So just by putting a lot of histological images as a training set in the deep learning algorithm, it could very accurately predict by giving the new input set as whether it was a normal histological set or a cancerous. Even the radiograph, the chest X-ray, the machine learning tool was very good in predicting whether the radiograph was a normal or it was a pathological radiograph. So next slide. Yes, so it is from the same paper. It's just a fraction of that. The first of these algorithm is one of the multiple existing examples of an algorithm that outperforms clinical doctors in image classification tasks. In the fall of 2018, researchers at Seoul National University Hospital and College of Medicine developed an AI algorithm called DLAD, that is Deep Learning Based Automatic Detection. Deep learning is a more advanced feature of machine learning. So they developed an algorithm called DLAD to analyze the chest radiographs and detect abnormal cell growth such as potential cancer. The algorithm's performance was compared to multiple physicians' detection abilities on the same image and outperformed 17 out of 18 doctors. So you can see the accuracy of this AI-based model. It predicted more accurately about the radiograph compared to 17 of the doctors. Only out of 18, only one could predict exactly what this AI-based algorithm predicted. So it is such a useful tool. So next slide. The second of these algorithms comes from the research from Google AI Healthcare. It was on news in 2018, who created a learning algorithm called LYNA, or Lymph Node Assistant. So lymph node is a very initial stage of a tumor. So LYNA analyzed histology slides, a stained tissue sample to identify metastatic breast cancer tumors from lymph node biopsy. and you can see in the last sentence the accuracy was 99%. So this from the same piece of information as the previous slide, but you can see the accuracy of AI predicted image in terms of diagnosis is almost 99%. Furthermore, when given to doctors to use in conjunction with the typical analysis of a stain tissue sample, LYNA halved the average slide review time. So using these kind of technology, the time consumed in diagnosis can be reduced to almost half. Okay, so this is an excerpt from an interview from it was an interview from the managing director of the Apollo Group of Hospitals, Sangeeta Reddy. So she said that artificial intelligence won't replace the doctors, but the doctors who don't use AI will be replaced by the doctors who are using AI. So it's not that computer system will completely replace the human beings, but it will be a huge advantage to the human beings who are using the AI, and those human beings will replace the humans or the doctors who are not using the AI. Though again, it all depends on the training. So whether it's a training of the human beings or the training of the computer algorithm, everything depends on the training. So next slide. Yes. Because I'm a huge fan of movie, I can't live without movie. So this is I hope everyone can recognize this song. Anyone who can't recognize, or anyone who can say which song is this? Okay, so it's just a analogy. This is a song from a film called Pardes, and the song is "Do Dil Mil Rahe Hain Magar Chupke Chupke." So I use this image because in this session, which we are currently into, biology and AI mil rahe hain magar chupke chupke, and I use this analogy for that. So next slide. So you can find numerous papers currently through Google whereby AI has been to the rescue for a lot of clinical and basic research applications. So next slide. Yes, again, a few of those examples. Okay, if you can see the picture on the top right-hand side, it is an article from Nature Biomedical Engineering. So, detection of signs of disease in external photographs of the eyes via deep learning. Interesting paper whereby AI model could predict just by the retinal scan whether the person will have a cardiac disease in the next 5 years or not. So you can see this is a completely interesting and very surprising thing, that just by giving as an input a lot of retinal scans to the AI algorithm, it was very accurately predicting whether the person will be having a cardiac disease in the next 5 years or not. So again, I'm repeating the same statement. Though I don't work at core with the AI, but AI is going to be the next revolution in anything related to biotechnology or clinical medicine. So next slide. Yeah, so that was the end of the slide. So anytime you can reach me at this particular email ID, and I'm out of all the social media, I'm active only on Twitter. So you can reach me at this Ekitilayan. Okay, thank you. Thank you so much, sir.
Thank you.
And
Thank you.
sorry to bother you.
It's open
Yes,
for question answer. Anyone have any question, please ask. And I have given you all the feedback form. So if you need certificate, you must fill that feedback form, which I have given after 40 minutes. So those who have joined after 40 minutes, they will not see that chat box feedback form. If you have any question, please ask question to a speaker. No question?
Hello, sir.
Yes.
Yes, sir. Sir, I cannot take the screenshot of email ID.
Okay. So he is writing in chat box, and you can
Yes,
connect
sir.
with me through LinkedIn. So I will tag him. He's
Can
writing in
I
chat
share
box.
the email ID? Yes, sir. So this is my email ID. If anyone of you wish to connect with me, I'm always there for science.
Sir, can we get the PPT?
So
sure.
You
Sir,
can watch video. I will upload this video on YouTube after his permission, Ashwini's permission.
Yes, sir. Yes, sir. Yes, sir. Sure, sir.
Then you can watch this.
Sir, thank you.
GPT will not
Thank you, sir.
take. Hello.
Thank you, sir.
Sir, I think there was a question.
Yes, please ask.
Yeah, it was from microbiology and background. Okay. So there was a question from Maqsood Ahmed. Request to share AI tools for microbiology and biotechnology scholars.
Yes,
Okay.
but
It was in my slide also. One of the most used tools currently based on AI is AlphaFold. So very recently, AlphaFold has tied up with UniProt also and EMBL, European Molecular Biology Limited. So because currently UniProt is one of the best place you can find out all the information about a protein. So recently, AlphaFold has done a MOU with UniProt, and it is going to predict almost all the structures. So if you can, I'll just share the name of the software. It's ALPHAFOLD, AlphaFold. If you can go to the website of AlphaFold, you can see the space where you can just put the sequence or the name of the protein, and you will get the predictor structure, the AlphaFold predictor structure. So that is one of the tool in silico medicine. As I said, all the three tools developed by in silico medicine, the PandaOmics or the Chemistry 42 or in Clinico, all those are there for licensing. So you can purchase those tools if you want to work on drug discovery. It is available through in silico medicine. Apart from that, there are a lot of things. In fact, AI is such a random thing that people who are working on machine learning, they are developing their own tools for their applications. So it is such a random thing. It's a kind of software programming, computer programming. So a lot of people who are working in machine learning based computational application, computational biology, they are also developing their own tools. So there are a lot of examples you can actually find. I hope I have answered this.
Sir, here I would like to just add one point what Ashwini sir has said. Earlier, as the condition was database, as was the condition for the database was there, that every other scientist who was involved in bioinformatics application and all those software developing things, they were trying to look for the generation of databases. In the same way, since the current science, that is more involving the machine learning, more involving the computational biology. So the person or the scientists who are involved in this field, they are now looking for developing the tools which are based on the AI. So as the science progress, the tools and the techniques, they also get advanced with the advancement that is going around to all of us. So in the same way, earlier the position of the database was now of the AI tools.
Exactly, ma'am. So now I hope everyone can be with me in the same page that gene sequencing is becoming very common. And there's a lot of changes in the gene, even in terms of SNPs or different mutations. So a single gene, even normally, can give rise to multiple proteins in the form of alternative splicing. But still, because of certain mutations, a single gene can have multiple proteins with different folded structures. And that's why going just with the gene sequence is not enough currently. So if there is some tools based on AI which can very accurately predict the correct structure of the protein or multiple proteins coming out of that particular single gene sequence, that will be a huge help in diagnosis and drug discovery. So yes.
Preet Janju is asking what is the accuracy of these AI tools.
What is accuracy? Okay. So I will again use one of the I'll again use one of the analogy. So we are all familiar with a very basic book of biochemistry called Leninger. So if someone has only read Leninger throughout his or her life, compared with a person who has thoroughly read Leninger, Stryer, Voigt and all other biochemistry books, so who will have a better knowledge of biochemistry? So the accuracy of AI is always based on the kind of training we have given to the algorithm. More and more training we have given, more accurately it can predict what we want. So it's always based on the training. That's all, and that's why it is called machine learning. We are trying to teach the machine something so that we can get the required output. That's all.
Hello, sir.
Yes, कृषि.
Yes, Krishi.
आवाज नहीं आ रही तुम्हारी, जोर से बोलो कृषि.
Hello, there was one slide about CAR T technology. So could you please tell me some
Yes.
more things about it?
Okay, so that will be a topic completely beyond this particular session. It's not about AI, but yeah, still we can have a bit of AI in that also, but in a very nutshell. So I hope you have heard of T cells, one of the immune cells in our body. So
Yes.
one of the very basic function of T cell is to find out any damaged cell and then kill it directly. Okay, so in CAR T, the full form of CAR T cell is chimeric antigen receptor T cell. So what happens, every second in our body, a normal cell is getting converted to tumor cell. But because of the normally functioning immune system, the immune system kills the tumor. But why a tumor becomes a tumor is one of the reasons is that it escapes the immune system. And there are a lot of ways
System does not work.
which are tumor system. Immune system works effectively. It escapes the immune system. So in Hindi, वो ट्यूमर सेल इम्यून सिस्टम को बेवकूफ बना देता है. One of the things it does is to shield it, to create a shield around itself so that the immune cell, tumor cell, or it creates a shield that the tumor cell, the T cell, consider the tumor cell as a normal cell. So when it considers the tumor cell as a normal cell, it is not going to kill the tumor cell. So this is the kind of shield it creates. And therefore, in every tumor, literally every tumor, there are a lot of T cells in the tumor also. But they are just sitting there. They are not able to kill the tumor. And the tumor is still able to grow size. So what happens in the CAR T cell, we take out the T cell from really the
आपका वो हो जा रहा है, आवाज कट जा रहा है.
Okay, sir. Okay, so I'll again repeat.
Last.
So when a tumor cell creates a shield around itself, the immune cell is not able to see that, or moreover, the immune system sees that it's not a tumor cell; it's a normal cell. So in CAR T cell therapy, what we do, we take out the tumor cell. We take out the T cell from the patient itself. We modify the T cell in the lab through recombinant technology. We put a new protein in the T cell, and that protein is expressed on the surface of the T cell. Now, this is a completely engineered cell. Now, this recombinant protein is able to find out and bind to some specific proteins present on the surface of the tumor cell. Now, when this binding occurs, the engineered T cells are very well equipped to kill the tumor cell. Now, when these engineered T cells are made, they are grown in the lab. They are put in the environment where they can replicate, they can divide, and then we have a sufficient number of engineered T cells.
These should attend CRISPR technology. That is one of the CRISPR technology for T cell.
Yes, sir. Yes, sir.
So this is part of genome editing, है ना?
Right.
हाँ, वो इम्यूनोलॉजी में इंटरेस्ट है इसे.
आवाज नहीं आ रहा. तुम कहाँ से बोल रहे हो?
हाँ, वो इम्यूनोलॉजी का ही टॉपिक रिलेटेड है ना, तो
Right.
So आजकल एक ही टॉपिक लिमिटेड नहीं होता है, उसमें मल्टीपल टॉपिक्स इन्वॉल्व होते हैं, है ना, इंटरडिसिप्लिनरी.
यस सर, थैंक यू सो मच सर.
इम्यूनोलॉजी में जेनेटिक इंजीनियरिंग लगा हुआ है कृषि.
हाँ सर, रिकॉम्बिनेंट टेक्नोलॉजी भी.
Yes, this is asking, does bioinformatics involve AI? अरे भाई पूरा bioinformatics
Exactly.
doing blast and other things, they all are based on the learning जो उसको teach किया जा रहा है, उसमें जो बताया जा रहा है, that is bioinformatics is completely that. क्या मैडम, पूनम मैडम बताएंगे.
यस सर, एक्चुअली अभी क्या हो रहा है कि पहले क्या था कि सॉफ्टवेयर प्रिडिक्शन, मशीन लर्निंग, लैंग्वेज सबका यूज करके आप प्रिडिक्शंस और जीन के बारे में पता करते थे। अभी एआई की वजह से, जैसे अभी एक एग्जांपल सर ने दिया था अल्फाफोल्ड का। बिलीव मी, लास्ट जब अभी मेरा एक कोर्स चल रहा था बायोमेटिक्स का, स्ट्रक्चर प्रिडिक्शन का, उसमें एक टॉपिक था। I told everyone, and we have some example. For example, one protein that I have worked with during my PhD. We have the domain structure of that protein, but what you can do with AlphaFold, you can have the complete structure of the protein. This of having the information whether in real experimentation or X-ray crystallography or NMR, what we use for the protein structure prediction, they can predict the smaller regions of the protein. But AlphaFold, that is one software, if the certain amino acid sequence, complete protein sequence, it can predict you the complete structure of the protein. You can derive different information from that structure. You can look for how many interactions are there, whether the amino acids who are there, they are present in the inner side, outer side, hydrophobic interaction, disulfide bridges, how it will be functioning against a kind of chemical agent or mutagens or anything that you can predict using those AlphaFold, using that AlphaFold software. So currently AI का रोल, जैसे अभी एक सिंपल सी चीज सब बोल रहे हैं ना कि AI will take the multiple jobs, but that would require the human intervention also. Exactly. Same thing is with the AI in bioinformatics. Even though we have the software, but how that software has to be used, AI can just give you the result, but how the implication of that AI software has to be done for the real science or for the output, that would be only dictated by the human intervention. So the correct answer. We can use software and use the AI tool that may not give you the accurate thing that you are looking for. That is also based on the assumptions or the hypothesis which we use AI tool. That would be important. So no doubt AI का intervention बायोइन्फॉर्मेटिक्स में भी start हो चुका है, बायोटेक्नोलॉजी में भी start हो चुका है, but actual उसका जो inference है या आप उससे क्या decipher करते हो, वो depend करेगा आपकी hypothesis के साथ में कि आप कहाँ शुरू किए थे और उसका क्या ultimate result आप चाहते हो. तो no doubt AI has turned into bioinformatics bayotechnology. अगर आप देखो तो that has to involve all those computation because AI itself. So अगर आप बायोइन्फॉर्मेटिक्स में AI का यूज नहीं करते हो, आपकी चीजों को काफी इजी भी कर रहा है, नो डाउट. कोई भी स्ट्रक्चर, जैसे एक सिंपल सा ही अभी हम लोगों ने बात की, स्ट्रक्चर कंप्लीट प्रोटीन का निकालना एक्सट्राग्राम से बहुत डिफिकल्ट है, बट AI is helping you to get the structure. भले ही आपको सिमुलेशंस करने हैं, भले ही आपको ये चेक करना है what structure you are getting, is it work according to your hypothesis, is it actually working in that same line that you are thinking, or if not, then what should be the changes that you may require. So no doubt, obviously AI का रोल तो है, mathematics and
And yes, and just as another example, protein is always protein always has a solvation layer of water. So, and in XRD, before NMR, when XRD was the only look in the structure, about 15-20 years back, it took almost 2 to 3 years of dedicated time to get a good crystal of protein.
Yes.
Yes.
Single
Exactly. Exactly. So it took a complete PhD to go through to get a bit of good structure of a protein. So it was a task. And with AI, we can just have a structure within 2 minutes. Just through software. Okay, so selection is again a very broad domain. I'll just have an example of one of this domain, which is called pharmacogenomics. Though the term pharmacogenomics came much earlier than the term personalized medicine came, but one very common way of understanding pharmacogenomics is that one drug in a single dose will have different effect on different people. Right, like paracetamol 650 mg can have different effect on different people. Like in some people, it can bring the fever in 30 minutes. In other, they can still wait for 2 hours to get the fever down. So it's not about only the drug that we are giving inside the body. It's also about how the body is acting upon the drug, because that is the most crucial thing. So, in terms of in the domain of pharmacogenomics, the thing is how the body is going to act upon the drug. So, in terms of drug metabolism, a single enzyme can have different folded structure and different sort of a range of function in different individuals. Now, to find out that disturbed function of that particular metabolic enzyme that is metabolizing the drug, we need to have these softwares or tools, AI tools, so that we can predict in out of 5 individuals, A, B, C, D, E, not everyone is going to require the 650 mg of paracetamol or any other drug that is
so let her first go through the precision medicine talk. We have already organized. Shruti, you can take a lecture from me. I have not uploaded, but you can take. We have organized precision medicine.
Yes,
then you can integrate it with artificial intelligence. Yes. So first go through that lecture. Any other have any question related to today's session? Any suggestion you can write in chat this form, Google form, feedback form. If any other participants have any question, please ask. Otherwise, we will wind up. We are going beyond the time. कुछ questions नहीं है? If there is no more question, then from my side, thank you very much, Ashwini ji, for sparing your time.
Thank you so much.
We are trying to disseminate knowledge. Since we are easily available, so people are not joining, and few people they join only for the sake of certificate. That's why we don't give them this Google feedback form earlier, and also not after this lecture in between. So that the people who joined after 40 minutes, they will not access that Google form. And those who are joining and leaving, they will also not access. So we randomly give after 30-40 minutes of the lecture. So that my motto is, our motto is to disseminate knowledge, not only to give you certificate and keep on compiling that certificate. You must watch, there are so many lectures on advanced topic, you will not find anywhere lectures on that topic, and we are not also giving you 5 minutes, 10 minutes. So many people suggest me that you split these videos, so essence of that lecture will go. So we are not splitting it. If you have patience, you watch complete lecture. So many lectures available on advanced topic on my YouTube, and you keep in touch to attend. So we will again organize several lectures. So next tomorrow is also on apoptosis and cancer pathogenesis on 16, single cell omics. So you keep in touch, watch. You should learn, and you should also disseminate knowledge in future to your colleague, your juniors. That is our purpose. Poonam madam, officially, yes.
If you allow me, I'll just like to add what sir has said, just in addition to that, for all the students who are currently with us, science need a lot of dedication, and this is true for all aspects of science, whether biology, chemistry, physics, anything, or any sort of research, even through technology, engineering. It needs a lot of dedication, and we have a very good example currently with us, Gopal sir. I know for last 4 years, right from the time we were struck into the first lockdown, sir started a very beautiful series on recombinant DNA technology. It is a 11-day course, and I can personally vouch that this RDT course is the best you can find out. Not because I know Gopal sir personally, because I know the kind of effort he puts in. So besides all the teaching and research he's involved in, he's doing these things, different sort of teaching through online mode, whether it's a recombinant DNA course or a genomic proteomics course, or we have just started this science dialogue for last 4 or 5 months. He has been involved actively in all these things. So this is another level of dedication that he has, and this is something we can learn a lot from him. So science doesn't only need dedication in lab or in classroom, it requires dedication at all the levels. And thank you, Gopal sir, for this initiative.
Thank you for supporting Ashwini ji, Poonam madam, so many our colleagues and juniors, they are dedicating time on my behalf for dissemination of knowledge. So that is also my achievement. In this 4 year, we have gained confidence and trust of so many researchers like you people. Poonam madam is also conducting now bioinformatics course in 200 rupees only. And I try to bring all the talented people in one basket. So only those who have confidence, because you are not knowing who are out another side. You are not knowing who are participants. Sometimes directors, deans. So that needs also dare to deliver on any topic. So many scientists and researchers are requesting me to give me opportunity, but when we say them key participants are from diverse background, so they then deny, they don't dare to speak, because they are generally they ask कि BSc का student होगा कि MSc का होगा कि क्या रहेगा सर, किस टाइप के रहेंगे. Generally delivering in closed room, that is easy, but delivering openly where you are not knowing the participant, that needs more dareness. So thank you for joining us, and I request those who are confident, those who are teacher and confident on any topic, I can provide you platform so that you can disseminate knowledge, but you must be confident on your topic. That is required for online teaching. It's not easy in closed room कि 20-25 स्टूडेंट हमारे हैं और उसको हमने टीच कर लिया. यहाँ पर मैं पूरा ओपन है. And we are taking live. This is also not recorded. So anyone can ask question in given topic. जो भी topic है. So thank you very much.
Thank you, sir. Thank you so much, sir. Thank you so much.
Madam, कुछ बोल.
Thank you so much, sir. And Ashwini sir, thanks a lot. Again, I told everyone in the first session or introductory part that I have already attended one of the lectures from Ashwini sir that was on shoe engineering. And I can assure you, any of the topic that is going to be discussed in this platform, that is going to benefit you, because such kind of knowledge in 1 hour, it is very difficult to get in that. And that too, the orator or the speaker, he or she is completely involved in this seminar or in this kind of lecture series. So thank you so much, Ashwini sir, for elaborating us on the AI role in this biotechnology field. And again, thank you, Gopal Jee sir, for your effort. हाँ?
जब भी बोलते हैं आप आ जाते हैं, so thank you very much.
अरे नहीं,
Small key.
Sir, all credit goes to you, sir. वो कहते हैं कि
association का role
माला में जो धागे का रोल होता है ना सभी को पिरोने का.
Yes, sir.
Friends who has not attended engineering lecture, you can watch his video on my YouTube channel. You can take from me also or in my YouTube channel. He has a very good, wonderfully delivered on tissue engineering also. You can lecture on bioinformatics, basic bioinformatics delivered by Poonam madam, and there are so many lectures available. So I request you all to visit and watch. Thank you very much, everybody. Good night.
Okay, thank you, sir. Thank you so much.
Thank you, sir. Thank you, ma'am.
Thank you, sir.